Now, this is going to be a fairly technical discussion. Let's get started and go through the agenda. So today, what we want to do is give you a little bit of an introduction about Nebius. Then we'll talk about the challenges that we've seen in the bioinformatics industry in general, how GPUs can be used to improve that. We'll talk about some of the work that Arseniy has been working on, and we'll make it interactive. Then we'll talk about why Nebius is a perfect choice for a lot of the computational work that Arseniy has been working on. And we'll talk about what next, and we'll finish off with a Q&A session just at the end. Let me start off by telling you about Nebius, since none of you are using Nebius at the moment. Let's see. As you know, we are a GPU cloud provider. We are quite unique in the sense that we are NASDAQ traded, NASDAQ listed for a neo cloud. That's quite an achievement. We've raised a significant amount of money from investors, including NVIDIA and Archaeal in December. We are an NVIDIA preferred partner. We have dedicated teams of AI, machine learning engineers and scientists. There's fun fact, we have more than 1.3 kilometers of optical cables in our data centers, where we have a lot of the infrastructure. And we're working on getting a lot more patents, but we've so far applied for 41 different patent applications. 6 of them have been issued. We are also working a lot on open source environments as well. There's a lot of GitHub repositories that we offer in terms of the software and also the hardware side of what we do. Just to give you an understanding of our global presence and the momentum that we have data centers listed here in Finland, in France, in Iceland, and 2 places in the US. We have a number of engineering hubs positioned all over the world. But this is just to show you how much of a global company we are and the momentum that we have for the AI industry. In terms of what we offer the full control over the value chain. So it's a full stack solution where not only do we offer the data centers and manage the hardware side of things ourselves, but we also have the cloud platform mainly for GPU training and the AI studio for all of the inference needs. In terms of life science and healthcare, what we are doing and what we're working on in this vertical is split into these 3 main segments. That's life sciences and drug discovery. That's genomics and multiomics, and then health tech. Everything to do with the MRIs, CT imaging, but also various tools and wearables that might be associated within the clinical environment. In terms of the packages and things that we offer at the moment, on the left, you'll see part of our training solution, that is the GPU cloud, with the dashboard and full stack solution that it comes with. And on the right, you'll see Nebius AI Studio, which is for all of the inference need. And that's quite a lot of excitement on that as well. In terms of inference, we not only offer access to the most common open source models like Meta's Llama and Mistral and even DeepSeek, but we also offer a number of healthcare and life science related models as well, such as Biomed LLM, M42 LLM, and others. So we're quite excited to be able to offer very dedicated life science and healthcare LLMs. In terms of what we're going to be talking about today, we've got something special for you. We want to make sure that we are able to go through and share with you how a technology originally built for gaming is revolutionizing the way that we decode life's mysteries in bioinformatics. So if we dive in, we know that when we're talking about CPUs and bioinformatics, there are a lot of tools that already exist for CPUs, like BLAST and various R packages designed specifically for CPUs. We know that CPUs handle sequential tasks very well and complex logical irregular memory access very well. CPUs are ideal for text parsing, and quite a lot of bioinformaticians, especially those trained in biology, they prefer CPU environments and they are familiar with it. But what we are going to be talking about today is the role of GPUs within this and how we can have an evolution of the technology that we're using. At its core, it's about harnessing the power of GPUs. to not only work through the workload, but turbocharge the analysis of all biological data. Think of GPUs as an unsung hero of modern computing. So unlike traditional CPUs, which process tasks sequentially, GPUs can handle thousands of operations in parallel. In bioinformatics, where we're often swimming in oceans of data, DNA sequences, protein structures, genomics data sets, this is a game changer. There's already incredible GPU-ready tools available, such as NVIDIA's Clara Parabricks, which helps accelerate a lot of the tasks that you're facing. They offer massive parallel processing for data-heavy applications. Imagine that you're trying to align millions of short DNA reads to a reference genome. On a CPU, that might take hours or even days. With a GPU, you could be sipping a cup of coffee while the same work finishes within minutes. And that's the kind of acceleration we're talking about. Why GPUs are the future? A lot of people ask. Well, we would say, and we would summarize it by saying that biological data size and complexities are doubling rapidly. We're seeing huge exponential growth within this, and GPUs really unlock faster analysis. They're critical at time-sensitive research, particularly around personalized medicine and others. But it also allows GPUs to enable scaling solutions for a lot of population genomics and lots of studies that require high throughput. The key takeaway that I would say, if you get anything out of this lecture, is that CPUs dominate for now, but GPUs are the future as bioinformatics scales and processes improve. So how can GPU accelerated workflows really improve bioinformatics? In terms of cloud GPUs in particular, we see that GPUs have a massive effect on parallelism, reduced compute costs, faster data processing. We'd say that it improves machine learning and AI applications. So we've seen enhanced accelerations in molecular dynamics simulations and others. So why does this matter? I'd say three big reasons: speed, scalability, and costs. So first, speed. Tasks like sequencing and sequence alignment, in particular molecular dynamics, they can run 10 to 100 times faster on GPUs. Second, as I said, scalability. When GPUs let us tackle much bigger data sets without needing a room full of servers. And you don't need to have a dedicated space for this, especially when you're using cloud GPUs. And third, I would say cost effectiveness. So for labs or institutions with limited budgets, a single GPU workstation can deliver super computer-like performance at a fraction of the price, especially in the cloud like Nebius. You don't need to manage it yourself. You don't need to worry about the power supplies, the cooling, the infrastructure. All of that's taken care of. And I would say that for us, it's really important to go through how GPUs can be used to take you to the next level. So Arseniy, I know that you were using Nebius's GPUs to do just that. Could you please tell us a little bit about the work that you were doing and take us through a little bit of that?
Yeah, sure. So I think that the source of this research is just hype of CRISPR-Cas based technologies and the Nobel Prize that was awarded to the research behind genome editing. I thought that maybe we can find something similar to CRISPR-Cas, maybe not in bacteria, but in archaea. And during my experiments, I encountered a problem with CPU-based tools, especially with the time for computational experiments. And I thought that maybe there are some alternatives to common CPU-based tools. And yes, there are such alternatives. What's behind genome editing? I think that might be one of the most interesting cases in science because the history starts with pure research that was made by Japanese scientists in 1987. Actually, they didn't try to find CRISPR-Cas. Nobody knows that there are some sequences with such functionality. Instead, researchers were trying to identify gene responsible for conversion of alkaline isozyme in Escherichia coli. And the exciting thing for me personally here is that this pure biological research set a foundation for a genome editing toolkit that is now widespread in various of biotechnology applications. We now have different options for treatment genetic disease that are even FDA approved. And not only for medicine, we have applications based on CRISPR-Cas9. We have some options for agriculture and food industry, and it's just a powerful tool for other downstream analysis and research.
I'd love to understand why you used archaea in this going forward. What made it special? What was the decision behind that?
Yeah, apart from phages and viruses, we know that there are three main domains of life, including eukaryotes and bacteria and archaea that are prokaryotes. The initial work in which researchers found CRISPR sequences that now are widespread in biotechnology was found in Escherichia coli. It is a bacteria. But I think that archaea are also quite interesting from the perspective of research and potential new insights in this field. So why it is so? I explicitly want to highlight three points here. The first is archaea shares features of both molecular mechanisms, both eukaryotes and bacteria. The second one is that they live in extreme conditions, like hyper salty environment or environments with extremely high pressure. And it raises the question, so how do they adapt to these conditions? So they must have strong defense systems, maybe not only against microbes and phages, but in general. And the third thing, but also quite important here, is that archaea, compared to bacteria, are less represented in research. And it is primarily associated with the complexity of working with archaea. From the three superphylum and one phylum of archaea that we know now, only some of groups we're able to cultivate. But I think that we will find more insights in near future that will be made on top of archaeal research.
Wow, that's really interesting to see how it makes them so unique in that research and give you some really interesting findings. I'd love to understand how you've been working on this project and what you've been doing there. Could you take us through that?
Yeah, of course. So I've made quite simple pipeline combining different tools, and I ran it on both CPU and GPU. And I not just only made some results from like scientific point of view, but also compared performance gains on GPUs. So we start with fetching data from UniProt database. I just tried several different experiments and found out that we can find some proteins that are marked as associated with immunity, defense mechanisms, antimicrobial activity. But after that, exclude those proteins that are associated with CRISPR-Cas. We all know the widespread of CRISPR-Cas, but I believe that we'll find more interesting results from systems that are different, that are apart from CRISPR-Cas. That's why I excluded specifically those proteins. Next, I wanted to expand a data set, and I ran MM6 to find homologous proteins. And for resulting proteins, I generate embeddings leveraging ESM Cambrian model. And resulting embeddings, I clustered with k-means and plotted results using UMAP, utilizing RapidSQML. I think that the central part of this pipeline is quite good. But on top of that, I added some not really pure scientific step involved AI DeepSeek based annotation of clusters. So yeah, as a disclaimer, I don't think that all these pipeline might be seen as a pure research. It's like highlight of different tools that we can combine to make a pipeline and highlight GPU acceleration gains that we can achieve leveraging GPUs. And yeah, the resulting clusters I annotated using a DeepSeek reasoning model.
It's really good to see how you can combine multiple tools within that to make the process flow based on what you were using. Really interesting to see that. In terms of the difference between what you were doing in CPUs and GPUs, what kind of efficiency gains did you see?
Yeah, I think it is maybe the main point of today's webinar. On this slide, we can observe performance gains, especially for the main steps that I've done during my small research. And last autumn, initial authors of MM62 collaborated with researchers from NVIDIA to make it possible to run it on GPU. And they made an impressive work. And now with an option for GPU run, we can achieve more than 4 times faster execution for homology search. And maybe for archaeal, absolute values are not so big, even for CPU run. But think about bacterial research. Current pipeline might be easily translated to bacteria, and there will be millions of sequences. So this cut off of execution time will be really significant. The next step is making embeddings utilizing ESN Cambrian model. Here is just for the sake of interest and to make this 2x2 plot, because we all understand that PyTorch model is not intended to be run on CPU in general, but yeah, we also can observe quite impressive decrease in time on GPUs. But one of the most interesting thing here, I think, is UMAP. More than 20 times faster on GPU. And also here we can see absolute values and relative numbers. And we need to concentrate on relative numbers because once we will try to translate this research to bacteria or making some more complex analysis, this decrease in computational time will be definitely significant. So in total, we achieved 12 times faster overall run of GPUs.
So it looks like you can do 12 times much work in that time.
Yeah.
How was GPU acceleration enabled in general? What did you do to make sure that it was giving you the most of what you were doing?
In the subsequent 3 slides, I'll highlight just a few parts of the code that are available on GitHub, but those parts that I want to explicitly mention. So the first step of my pipeline includes MM62 homology search. And the interesting point that because NVIDIA helped with porting this tool to GPU, they made it possible to specify CUDA architecture. So because I ran it on Nebius, I was able to run these experiments on Hopper GPU H100, which is architecture 9.0 in CUDA nomenclature. So we can specifically highlight that now. Let's enable CUDA architecture 90. And yeah, just remember to use GPU flag in your actual search. It's one of the main points here. The second step is ESM Cambrian. It's quite simple to use. It's similar to, it is not just Hugging Face transformers, but it has similar API. And just don't forget to put it on CUDA. And another thing that I want to highlight here is that let's use polars, not pandas. And the last thing is QML. So we can run k-means and UMAP on scikit-learn. And also there is an option for QML. Because I did this research before NVIDIA conference, GTC, I needed to explicitly call QML cluster k-means, QML manifold UMAP, to make it possible to run it on GPU. But developers from Rapids present a new feature in QML that is now publicly available, that now you can just write one line of code load extension in your Jupyter notebook, and it will transform under the hood all your scikit-learn code into QML and run it on GPU. So if you have some scikit-learn Python notebook or just Python module, zero code changes, and you can run it on GPUs, just Google it, like zero code change for QML and scikit-learn conversion. Really convenient thing for current code base.
I saw that Shaquille Rafi in the audience. He said, "Yay, polars!" Really happy about that. In terms of what you were doing, Arseniy, what results did you achieve other than the increase in speed and everything else?
Yeah, results are promising. I'm not sure that all the subsequent analysis is absolutely correct, but it's just thought-provoking. So I made a dimensionality reduction using QMap and clustered points that are actually embeddings of sequences using k-means. And one thing that piqued my interest is unequal distribution of unannotated proteins within each cluster. So recall that I started with UniProt sequences from UniProt database. All of these sequences contain column with description. Some of proteins have description, others don't. And it might be like hypothetical protein or domain of unknown function and so on. And something like null hypothesis is that these unannotated proteins should be distributed uniformly. But here instead, we observe the case when more than 1/3 of proteins within one cluster don't have any annotation. And it raises the question, why it is so? To perform downstream analysis, I utilized Nebius AI Studio or running DeepSeek model. So with prompt that you can observe on the slide. And I asked model DeepSeek v3 to summarize clusters, to summarize proteins within each cluster. Most of the clusters are MGMT family proteins that are not an interest for us, but specifically the fourth cluster that is the most unannotated, also as well as 10th, might be something interesting. So we can observe that it contains halocene proteins, and also it contains PV and arginate domains. Both of them are known to be involved in some defense mechanisms. But I just Googled halocene encodes and PV and arginate encodes in Google, and I found no publications about it. And here we can observe that they are collocated at least based on their embeddings. It might be just a coincidence, or it might be something new, really interesting for downstream analysis. Also, 10th cluster is potentially interesting because it contains immunoglobulin-like proteins and LAMG. But yeah, fourth is the most intriguing here. And what I did next is to run new AI prompt. I asked, in this case, reasoning DeepSeek model, R1, to summarize results produced from the previous step. Here we have 3 different options for archaeal immune system architecture. I'm not sure that all of these are 100% correct and the model isn't hallucinating, but at least the second slide that represent what DeepSeek said us about potentially new defense mechanisms, it might be really working. So we know that arginate and PV proteins might be guided by some RNA and DNA mechanisms, but maybe it is done, at least in some organisms, by hallucins. Who knows? And the necessary part to understand whether we just reading hallucinations of AI model or not is just to check it in lab. So suggested some experiment validation steps. And a part of these experiments, I think that another one can be just find, proteins in prokaryotes are translated using operon system. And specifically for immunity, we know immunity islands. And we can find those organisms in which proteins for genes for PV and arginate proteins are co-located with hallucins. If so, it will be a good sign for us that even an immunity island may be checked with some neighbors of these proteins and find organisms with this collocation. And the best option here is that if these organisms might be cultivated in laboratory. And if so, we can just knock out one, for example, PV, and check whether hallucin works, or vice versa, knock out hallucins and check that PV and arginate proteins work. So it might be a good subsequent validation for wet lab, at least from my non-wet experiments, not like a biologist. I think it might be good subsequent analysis.
I love how the more research done to find answers, the more questions that appear. And it's fantastic to see that despite a lot of the work that was going on, there's still a lot of directions that this work is going to take you. It's incredible. In terms of Nebius as a platform, what would you say makes it a perfect fit for what you were trying to achieve and the goals that you were going for?
I think that with help of Nebius, I was able to run these experiments with the really great execution time drops. Yeah, with the more than 10 times faster with help of H100. And because I'm like a PhD student, I'm able to apply for a Nebius research credit program. So we provide to students and postdocs and in general researchers all over the world credits for the most recent GPUs, and you can obtain up to 8 H100 for like a year for your research. And yeah, I think it is one of the main part for this webinar, because most of participants are from academia. And also, we are not just a bare metal cloud. We have a lot of managed solutions. One of them are Jupyter Hub, so you can run your notebooks with just one click of your mouse on our website. And also, I know that a lot of solutions are made for Slurm, and we have a managed solution for it. Actually, it's Slurm operator for Kubernetes, but for you don't need to change your Slurm scripts. Just run it on our almost infinitely scalable clusters and run your docking or MM62 on hundreds of GPUs easily.
Yeah, that's a really good point as well. In terms of what's next, Arseniy, what have you got in mind for what you were doing?
I can suggest just one option out of, I don't know, millions of different subsequent analysis paths. So apart from option for just go to laboratory and try cultivate archaea, we can even narrow down this research. So one option that I previously said is to find collocation of hallucins and arginate PV proteins. And another is try to run the same experiment for bacteria. If you're not interested in archaea, the same pipeline might be easily run for bacteria. And with more data, you can create better understanding of the idea behind non-CRISPR defense and immunity mechanisms. And one option that I can suggest is to look at protein interactions. There are many options for prediction it, and maybe the last one that I used is from made by guys from Beamwell called APT, I think, and it's quite fast tool to just run it for millions of proteins. Next, we can generate a network, and we can start with convenient network X code, and with help of rapid investment of in zero code change, translate it under the hood to Q graph, and ran experiments on GPUs with no code changes, and just writing code in a convenient and familiar network X. And maybe we'll find the connection between different clusters. So as you have seen, there is not only cluster 4, but also 10th cluster that is also might be interested from the perspective of potential new defense mechanisms in archaea. So maybe they are interacting with each other. So yeah, there are a lot of options, just one of them.
We had a question literally just now asking us if we can share the GitHub repo. Is that something that you'll be able to do? Like, how could others use your work?
Yeah, I think it is available on, the link is on blog post page. I tried to make README as detailed as possible, how to install NM6. And actually, I used Marimo instead Jupyter, just because I like Marimo. I also included a guide how to install Marimo. And also I used UV instead of regular pip, and also added a few lines of code how to install UV. So yeah, there are 2 folders for CPU run and for GPU run. Both of them include the README for installation process. So yeah.
Thank you very much, Arseniy, for the session. It's been really interesting to get to know what you were working on, and how the power of GPUs can be used for all of this.